So we're interested in genome sequencing. So in order to give a bit of motivation, I want to say that genome sequencing is actually an important problem. It's a fundamental step you'd have to do in any modern biology problem before you can even start any analysis or start understanding any system. So it's sort of like computational power in the sense that you can never have enough of it. There's a lot of cells out there and a lot of species out there, and there's way more than we can possibly hope to sequence. Ideally, you would sequence every cell in your body, for example, if you wanted to understand cancer or the differentiations between different cells and so on. So just as a backdrop, you want to be able to do DNA sequencing fast and efficiently and cheaply. Okay, so what's currently done? Well, let's think of, here's our genome. It's composed of A, C, T's, and G's, and it's quite long. Our human genome is 3 billion letters long, 3 billion bases. Now, ideally, you would just start at the beginning and just read it from beginning to end. Now, current technology doesn't allow you to do that, so what's done is something called shotgun sequencing, and what that means is you get small fragments. Okay, so you get many copies of this chromosome, for example, and then you break it up into tiny fragments and you read each fragment. So here in this schematic, you get each of these is called a read, and it's of length about 100 or 500. It's a parameter that depends on the technology. And there's a lot of these. So there's 100 million, approximately, maybe 200 million, some number. And given all these reads, you stitch them together in order to reconstruct the original DNA sequence. All right, so what's the current state of affairs? Why is there any need to do anything new? Well, okay, so first of all, there's a lot of different machines. There's 5 or 10 different manufacturers that make these sequencing machines. Each one has different parameters, and then there are different algorithms used for each machine that mainly use heuristics and so on. So you would think, well, why isn't there one best algorithm? Why are there like 10 or 20 or 30 different algorithms? There should just be one best one, and why isn't there one best machine? I mean, it should just be one optimal. Okay, so this is sort of like the scenario that Shannon saw before Shannon came along and kind of gave a good framework for communication. There was the telegraph, and there was this, and there was underwater cables, and people did something totally ad hoc each time. So inspired by Shannon, we make an analogy with communication, and we say, well, okay, so you have this genome, and we want to know what is the minimum number of reads that you need in order to reconstruct it, okay? And of course, and this is with high probability, so probability approaching 1 for getting the thing correct. Right, so the basic question, just like a channel or a communication rate is the number of bits you get per channel use. Here we're interested in the number of nucleotides you get, so there's G of them, per channel use. And there's N reads, so we think of getting a read as a channel use. Okay, so what we're interested in is G over N, and we think of this as our rate. So just as Shannon modeled the source of the information that was then to be communicated across the channel in a statistical way, we're going to assume that there's a statistical model on the DNA sequence. So here there's some probability distribution on the DNA sequence, and similarly there's some probabilistic model for how you're getting these reads, just like there's a channel model in information theory. So that's kind of the basic schematic, and it's analogous to Shannon's picture, except that you can't control the source, right? You can't control the encoding. In communication, the encoding is part of the design. The engineer designs it. Here, well, the encoding has already been done. It's nature chose it. So all you can do is choose the decoder. How do I reconstruct the original sequence using the reads? I'll just give a sketch of an overview of the basic result for the most basic model that you can imagine. Well, what's the most basic model? Let's assume that this sequence here is IID, so each letter is independent and identically distributed according to some distribution P. So P here is the per letter distribution on the A, C, T, and G. So you fix that. The model we're going to assume on the reads is that each read comes uniformly at random and it's noiseless. Okay, so the simplest possible. And we're going to ask the question, well, what's the capacity? What's the best rate that I can get for a given choice of parameter? So a given choice of this length, L. What's the best rate that I can get? Okay, so it turns out that L actually needs to be normalized by log G. So L bar is the correct parameterization. So we're going to fix L bar, and we're going to ask, what is the best rate? Well, let's think about outer bounds. So what is going to prevent us from reconstructing the original sequence? All right, so, well, okay, as a first order thing, obviously if I have a gap, so I'm covering this part and I'm covering this part and I'm not covering this part, then these, for example, could be in a different order, and I have no idea. And so, okay, so coverage is an obvious condition for reconstruction, and that actually gives you this line. So we're below this line. Capacity has to be below this line. Now, another condition that's a little bit more subtle for reconstruction is duplication. So if I have, this little figure shows, if I have a duplication that's longer than the length of my reads, then in some sense, as I get here, I'm sort of lost. I no longer know how to connect this piece and this piece to this piece, right, in the correct order. And so actually, it turns out it's a little more complicated. There's, if I have interleaved duplications, that's what prevents me from reconstruction. But the probability of them is asymptotically the same, of having one duplication or many. So that actually turns out that the duplications are going to happen of the length of the reads if my L bar is to the left of this 2 over some Rényi entropy, so-called parameter. And what this says is that the capacity is 0 to the left of here. So no matter how many reads I have, I can't reconstruct. Okay, so this is the capacity curve. And that's, so far I've described the outer bound. It turns out, kind of surprisingly, that a very simple algorithm is optimal and that achieves this green region. And that's this greedy algorithm. So what is the greedy algorithm? It's very simple. It's kind of been well known in the bioinformatics community. It just says, well, I'm going to merge these reads one by one. So I'm going to connect this read to this read if it appears to have an overlap. And the way I'm going to do this is I'm going to start with the reads that appear to have the highest overlap. Okay, and the reason for this is kind of natural. Well, 2 reads that overlap extremely high, they're likely to actually be from the same place, because it's unlikely that by random chance some other read appears to have that portion. Now, as I'm performing these mergings, fewer and fewer reads remain. So at the very end, there's very few reads remaining, and so even though they're overlapping by a much smaller amount, there's fewer reads that can cause confusion. So it turns out that this greedy algorithm is optimal. Okay, and that's basically everything in a nutshell. I'll just mention that there's extensions to more complicated models that include noise in the read process, correlation in the DNA sequence, for example, a Markov model for the DNA sequence, and then still more complicated things that are actually important in practice, such as long repeats in the DNA sequence. It turns out that half of human DNA is composed of repeats, and you'd like to be able to actually overcome that. So the ID model is not at all accurate. So anyway, thank you very much for your attention.